Okay, so thank you very much, and thanks to Isa for organizing again this ABCnet session. And so today's lecture will address some basic principles in prebiotic chemistry. Before life could arise on a planet, the atoms have to organize themselves into very complex structures such as proteins or RNA molecules. Such molecules can't appear at once on the planet. They are the results of many complex organic syntheses involving many compounds. And today I will mainly focus on the first stage of the evolution of those compounds. At some point, at some as yet undefined stage of evolution, a giant leap is made and chemistry turns into biology. But I am a chemist and I would define myself as an astrochemist. So I will mainly focus on the first steps, and I will try to show you why astrochemistry somehow is linked to the origin of life on Earth, and origin of life on other planets.
Sorry, I'm in the middle of a ABCnet.
These are the outlines of my talk today. So first, I will talk about the chemical ingredients in a prebiotic soap. So for most of you, probably these are things that you already know, but some of you are not familiar with the chemical ingredients of life. So I will go in some details about this to get a clear view for everybody about the ingredients required for the origin of life. And then I will go back a little bit in time to give you some milestone in prebiotic chemistry to show you how the chemists are working on these questions. And then we will address the two kind of sources that can be investigated for the origin of the basic compounds for the origin of life, what we call endogenous sources, I mean the prebiotic compounds that can be synthesized on the Earth, and exogenous delivery of prebiotic compounds, so what can be the role of meteorites, comets in the origin of life. And then we'll go into some more details on chemistry and see some basic mechanisms to get from simple to complex, but not that complex compounds, as you will see, but at least to understand what are the first stage, the ones which are today well understood regarding the origin of life. So the first point of the outline, the chemical ingredients in a prebiotic soap. And we'll start by talking about life as a little robot. I like this definition, which is given by André Braque, which is a French chemist. And he has a very simple definition of life. He says that life is based on little robots making copies of themselves and capable of evolution. By evolution, is just talking about mistakes during the reproduction process. So starting with one robot and ingredients, the robot can make a copy of himself, and using the ingredients all around him, he can make other copies, and from time to time he makes a copy which is different from the previous one. And on this one you see that there is an evolution. The robot has two arms, so it's better. It's a better robot, so it's more efficient to make copies of himself. And for a chemist, and so there are many definitions of life, but for a chemist like I am, I'm quite happy with this definition, which is not talking about philosophy, not talking about complex process. It's just something efficient. One structure, one molecule which is able to make copies of itself could be considered as life, or at least a very good start for life. So if we look into how life as we know it on Earth is working, if we start to dismantle the living robots we all are, and we are that kind of robot. Bacteria are that kind of robot. We all know that we are made of at least one cell. This cell is made of ingredients, and in this cell you have molecules such as DNA, RNA, and other compounds named proteins. And proteins are made of amino acids, and actually the RNA is transmitting information from the DNA to build the actual proteins that we use in our metabolism. So if you consider the proteins are extremely complex compounds, but actually they are made of very simple ingredients. A protein chain is a chain of amino acids, as you can see here, and those amino acids are linked between themselves by peptide bonds. And you can imagine any kind of amino acid you can imagine, because an amino acid is a compound with a carbon atom, and on this molecule you must have at least one amino functional group, like here, and one acid carboxylic functional group, and then you can have a H here and any kind of other chain structure over there. So you can imagine a lot of different amino acids, but life is only using 20 amino acids, and if the R here is different than H, then the amino acid is asymmetric. It has what we call an asymmetric carbon, and we know that all the biological systems are using amino acids which are the same configuration, which is the L configuration. All the amino acids except glycine, because glycine has no asymmetric carbon. This is the question of chirality. I'm not going to dig into that today, but you must be aware that the origin of chirality is a critical question in the origin of life. So you have proteins. Proteins are extremely complex, but to build the proteins, you only need, only is a way of speaking, but you only need 20 different amino acids. And then if you look at the second family of very important compounds which are in the cells, you have the DNA and RNA molecules, and they are also extremely complex. But when you look at the pieces to build those complex structures, is not as difficult as it may appear. If you just consider at this structure shown here and look only at the colors, the blue parts are sugars. So on the DNA compounds, this blue sugar is a desoxyribose, and on the RNA molecules, this blue sugar would be the ribose. So those ribose are, or desoxyribose, are linked together by a small molecule, which is a phosphate, a P atom with 4 oxygen atoms around it, and those ribose and desoxyribose are linked with the phosphate, and on each ribose you have the nucleic bases or nucleobases, and you have only 4 different kinds of nucleobases on the DNA and another one for RNA. So in the end, to build a DNA compound, DNA molecule, you need ribose, 4 nucleic bases, and phosphate. So here you can see the details of the structure of the bricks leading to the DNA or RNA compounds. So it's, if you're not doing chemistry, you might be afraid by this formula, but in the end, it's relatively simple, and you don't need much to organize the molecules that are required for life. So as a summary to remind you what is necessary and what we are working on to build life and to understand the origin of life on the planet, or at least on Earth, we need to account for the synthesis in prebiotic conditions. I mean, without some chemist doing all the work on the lab bench, in prebiotic conditions which are relevant to the primitive Earth, which has an environment which has changed since the origin of life, and without any biological support. So you would need first to build membrane. So to build membrane, you need long organic chains which are synthesized. It means that one side of the chain likes to be in water and the other side of the chain doesn't like to be in water. So such compounds organize themselves into membrane, and you need to account the production of amino acids, and from those amino acids build proteins. You need to account the production of phosphate. You need to account the production of nucleobases. You need to account the production of sugar, and then link phosphate, nucleobases, and sugar to build DNA and RNA. So it's just like a Lego game. You need to understand the building of tiny organic blocks. So if you open a Lego box, you have many blocks, but some are blue, black, white, red, tiny, large. They have different shapes, different properties. And that's not the end of the story, because if you have amino acids, sugar, nucleobases, you need to organize themselves. And understanding the origin of life is making that kind of giant leap between these parts which are in the box and this kind of organization. And I will say that I'm not the one who built this town with Lego parts, but obviously someone did. And to understand the origin of life, you need to understand that kind of organization in natural conditions without any chemist. And that's a very difficult question to address, but I will try to show you the kind of progress that has been done in this field during the last decade and during also the last few years. So, some milestones now in prebiotic chemistry. You probably already have heard about Alexander Oparin, but I really like to quote his first book. He wrote his first book called The Origin of Life in 1924. And I really like this quote because actually everything is in his quote, and we are still working on his idea. In 1924, Alexander Oparin did not have an actual lab to test his ideas, but he was probably one of the first to connect astronomy, biology, chemistry, and he said that at first, I quote him, "At first we found carbon scattered in the form of separate atoms in the red hot stellar atmosphere. So that's nucleosynthesis. The atoms are produced, at least carbon, oxygen, nitrogen are produced in stars." And he said, "We then found it as a component of hydrocarbons which appeared on the surface of the Earth. So the atoms are organizing themselves into hydrocarbons. And in the water of the primitive oceans, this substance form more complex compounds, proteins." So at that time, he didn't know about the importance of DNA, RNA. So the biologists and the chemists were focusing on proteins. So he was saying that proteins and similar substances appeared. They acquired a more and more complex and improved structure and were finally transformed into primary living beings, the forebears of all life on Earth. And this is what we are doing, that we are still doing right now, doing what we call the chemical evolution. Before the classical Darwinian evolution, we were probably facing a chemical evolution, a competition between various reactions that would, in natural conditions, lead to the origin of life on Earth, because Earth has the environment that was suitable for the apparition of life. You've also all heard about the Miller-Urey experiment. So Miller-Urey experiment was actually the first time that the heparin ID was tested in the laboratory. And Stanley Miller ID was to test the kind of chemistry that would naturally be occurring on the primitive Earth. And he was facing a very difficult question because he had to decide to imagine what was the primitive Earth. And so he would imagine, it's simple to imagine that liquid water was on the primitive Earth, but he had to face the crucial question to decide what was the composition of the primitive Earth atmosphere. And he had to pick between various kind of composition that we would call oxidizing, which is the kind of atmosphere we have right now on Earth, made of carbon dioxide, nitrogen, water vapor, and dioxygen, or neutral atmosphere made of CO2, N2, water, and reducing atmospheres made of, for example, methane, ammonia, water vapor, and hydrogen. And based on observation of giant planets, the things which were commonly accepted in the 50s, he decided that most probably the primitive atmosphere of Earth was a reducing environment. So he simulated the Earth atmosphere in a bulb, in a glass bulb. He mixed methane, ammonia, water, and hydrogen, and this atmosphere was in interaction with the Earth oceans. So he had a second bulb in which there was liquid water, and he had a heat source because he thought that, and it was actually probably the case, the primitive oceans were warmer than they are right now. So if you take a bulb and if you put in that bulb methane, ammonia, water, hydrogen, nothing would happen. These are not compounds that we know that will react together and form more complex compounds. You need to add an energy source. And for the energy source, he could choose the photons of the sun, the UV photons of the sun, but at that time he had no efficient UV lamps that he could use in the laboratory. So he thought he would simulate thunder and lightning during the primitive Earth storms. And so he had two electrodes and he sparked a discharge into the gaseous mixture. And after a few days, this is how the bulb looks like. There is a brown deposit all around the bulb, and if he collects samples in the water simulating the primitive oceans, he had a surprise. Actually, he was hoping to get that kind of result, but it was really a surprise for most of the scientists at that time that a large range of organic compounds were synthesized, and he was able to analyze at that time that at least 4 amino acids were synthesized in the primitive ocean. So it was really considered at the time as a real breakthrough, because it was thought that if you add amino acids, then the next steps, I mean, building proteins from amino acids and building DNA, RNA would be something very, very easy to achieve. And if you look at the press release at that time, there were publications saying that scientists have understood the origin of life. But no, it was not the origin of life. It was under certain circumstances on a putative primitive Earth, understanding the synthesis of amino acids. And also there is a but. Here you see the brown coating on the bulb is a clear sign that there is a good efficiency of organic synthesis. The brown coating is carbonaceous matter. And if you change the starting material, the starting hypothesis from which you built the original atmosphere of the Earth, then you realize that you won't have the same efficiency in organic synthesis. If you mix methane, ammonia, water, and hydrogen, you have some kind of giant organic factory, and you get thousands and thousands. We know that there's a lot of organic, much more than the 4 amino acids detected by Miller in the 50s. You have thousands and thousands compounds which are synthesized during this experiment. But if you start with another kind of mixtures, a kind of neutral mixtures that nowadays we think are more relevant of what was probably the primitive Earth environment. If you mix CO2 and nitrogen, and if you spark a discharge, unless you have some control of the pH, unless you add some ingredient as antioxidant in the simulated ocean, then you get almost nothing, or at least something with very low, extremely low yields of production. So this Miller-Urey experiment was the starting point of what we now call the study of the prebiotic chemistry. But it's not the answer. It's not sure that this is what happened on the primitive Earth, because it depends on the actual composition of the Earth primitive atmosphere. But it's not the end of the story. It's the starting point of the story, and a lot of studies have been conducted after Miller experiment, and studies that were focused either on the production of organic compounds on the Earth, what we call the endogenous sources, or turning to space and focused on the delivery of organic compounds from extraterrestrial bodies like meteorites or comets. So I will review these sources, and I will start with the atmosphere. As I just told you, the original composition of Earth atmosphere was maybe not the best we could imagine to produce organic compounds, but we know that such environments do actually exist in the solar system. And if you see these pictures, most of you will be able to recognize what is this object in the solar system. This is Titan. Titan is the largest moon of Saturn, and here you have a picture of the atmosphere. And there's something going on in this atmosphere. There's something going on in this atmosphere because actually you can't see the surface of the planet. You can see the surface of Titan, and it's something that has no equivalent in the moons of either Jupiter, Saturn, Uranus, or Neptune. You are always able to see the surface, but you can't for Titan. You can't because there is a yellow haze hiding the surface, and this yellow haze is actually the result of the chemistry occurring in the atmosphere of Titan. And according to the starting material and the kind of energy deposition you do in this starting atmosphere, you have very different kind of results. If you look at this kind of starting mixture, which is the composition of a giant planet, if you spark electric discharge, if you do photolysis in such kind of atmosphere, you have a lot of organic chemistry going on. And if you look at the solid phase, when I place solid, in the table, it means that if you recover the solid material and if you put this solid material in liquid water, then after an hydrolysis, you would get amino acids, you would get nitrogated heterocycle. So you have a large amount of organic material which is synthesized. If you change here, you have carbon in the form of methane, ammonia in the form, nitrogen in the form of ammonia. And if you change ammonia into nitrogen, so this is relevant of the composition of the atmosphere of Titan, again, you have a lot of organic chemistry. And that kind of organic chemistry is simulated in the laboratory, and it results in the production of organic solid materials, which we call solins. And if you put those solins into liquid water, you get amino acid, you get nitrogated heterocycles. So nucleobases actually are nitrogated heterocycles. And if you get from those very reducing environments, such as the one I've just described, to environments with less methane and where the carbon is in the form of carbon monoxide or carbon dioxide, then you still have some synthesis, but less and less efficient, less and less diversity. And for environments which are more relevant to what we are expecting to be the one prevailing on the primitive Earth, you have almost nothing. And unless you get some drastic condition to get some synthesis done. So a lot of studies are conducted on this field. There are still a lot of questions, a lot of discussion on the actual composition of the primitive Earth atmosphere, and this is a very important point to study and to keep on studying. And exploring a moon like Titan will help us also to understand the kind of chemistry occurring in that kind of environment, because in the lab, you are in a lab. You can't make chemistry for billion years or even hundreds of years. You can do it in the laboratory, but you can study that kind of chemistry in the atmosphere of Titan. But on Titan, you can't expect to have liquid water, so you might not expect to have life on Titan. But Titan might learn us a lot about the kind of chemistry occurring in a reducing atmosphere, even if water is not stable at the surface. So, to summarize what I've just talked about the atmosphere, it is quite difficult to establish the real composition of the primitive Earth atmosphere. There have been estimations on the efficiency of synthesis depending on the redox state of the atmosphere, and the efficiency of synthesis can vary by several orders of magnitude. And if you try to calculate some kind of steady state concentration of organics in the primitive oceans, you can range from 0.4 gram per liter to 0.4 10 to the minus 3 gram per liter. So from quite concentrated organics in the primitive ocean to something quite diluted, from which you can't imagine to get a good efficiency of synthesis. So if we live for a while in the atmosphere and jump into the oceans of the Earth, we still can observe right now in the depth of oceans near the oceanic ridges that seawater can circulate through the crust. And in the crust, it is heated, it can exchange chemical with the rocks, it can interact with clays and minerals and have some kind of catalytic interaction with the minerals. And in those regions of the crust, there is somehow a kind of enrichment if reducing gases, and you have hydrogen, H2S, methane, and maybe possibly HCN and ammonia that can be produced in those environments. And then again, in the depth of the ocean, you have a reducing environment. You have large temperature ranges because at the exit of the hydrothermal vents, you can have temperature going up to 350 Celsius, and the water just around is only at 2 degrees Celsius. So that kind of dramatic change in temperature can be in favor of polymerization. But there is a counterpart, because high temperature also favors the destruction of the most fragile compounds. So it is a reducing environment, interesting for chemical synthesis, but the conditions in those environments are very hard and maybe too hard for the chemistry. But there have been studies, and people in the laboratory have been trying to simulate the chemistry of an hydrothermal vent. And so they start at high temperature, high pressure, they mix gaseous phase, aqueous phase, mineral phase, trying to reproduce the kind of environment you have in those hydrothermal vents. And here you can see a chromatogram that has been published quite a while ago in '92, showing that after that kind of simulation in the laboratory, you produce amino acids. So Stanley Miller simulated the atmosphere, and in reducing conduction, atmosphere produced amino acid and other compounds. And if you simulate the hydrothermal vents at the bottom of the ocean, in reducing environments, you also get interesting organic chemistry. But it's quite hard to simulate in the laboratory high temperature, high pressure. There have been studies in situ. People have been trying to collect organic material from those environments, but it's quite difficult because you may know, or at least you will learn during this ABCnet series of lectures, that life is able to adapt to a lot of environments. And of course, those hydrothermal vents are inhabited. There is life, there is bacteria that are able to sustain has rather high temperature. And if you analyze the organic composition of those environments, you see amino acids, but you see that their chiral distribution and their structural distribution are consistent with the biotic origin. So it's not, I mean, these are not amino acids that have been synthesized here, but they have been brought by life. So we don't know what happened in this environment when life was not yet established on the Earth, but today there is no abiogenic amino acid synthesis that has been found in those hydrothermal systems. But it's a track to follow, and people are still working on this track. And we now move to what we call the exogenous sources. And this is a very interesting track because actually it's not depending on hypothesis. It doesn't depend on what we thought the Earth was 4 billion years ago. We still collect meteorites which are falling on the Earth. We can still explore, observe comets, and such bodies are suspected to have remained unchanged since their formation 4.5 billion years ago. So what you get on Earth right now is actually what was falling on the Earth, probably with a higher flux. It's actually what was falling on the Earth when life appeared. So it's very interesting. And when you run analysis on meteorites, so today there is roughly 10 tons of meteorites falling on the Earth. But if you add to this quantity what we call micrometeorites, so it's hard to find a micrometeorite, but there are teams which are collecting them, trying to find them in the ices of Arctic and Antarctic. And if you study the composition of some of them, not all of them, some meteorites are made of iron, so these are not the ones interesting for the origin of life. But the kind of meteorites we call carbonaceous chondrite have an important content of carbon. So for carbonaceous chondrites called Ci or Cm, there is up to 3% of carbon in mass. And one of the most famous meteorite that has been studied in many laboratories is called the Murchison meteorite. And in this meteorite, more than 70 amino acids have been detected, including 8 amino acids which are used in life. So this is not what we call panspermia. Panspermia is the idea that there would be bacteria in the micrometeorite, meteorite, and falling on the earth, and you postpone the idea of the origin of those bacteria. So far there has never been any detection of bacteria or living system in a meteorite or micrometeorite. But there are organic compounds, and organic compounds such as amino acids which are actually detected. And this is a recent publication called High Molecular Diversity of Extraterrestrial Organic Matter in Murchison Meteorite, revealed 40 years after its fall. So it's a very interesting paper. If you are interested, I suggest you to read it, because those authors are using the most accurate techniques that are now available in the laboratory. And they have been able to extract the carbonaceous content of one sample of the Murchison meteorite, and they have detected more than 10,000 of different molecular structures. And they think, doing some statistics on their measurements, that there are likely millions of diverse structures in the Murchison meteorite. So the question of diversity or an organic source is not a critical question right now. We know that there is a high diversity of organic compounds in meteorite, probably a high diversity of organic compounds in comets. I will give you a few hints about why this can happen, why they may appear, they may be synthesized in comets and meteorites. So the question of diversity and origin of organic compounds is not the most relevant question right now. The most relevant question would be, what do we do with 1 million of compounds, and what are the interesting ones? What are the ones which have been actually involved in the origin of life? We don't maybe need 1 million of compounds, but maybe 1,000. I don't know. I have no idea, but it's a question that has to be addressed now. And I've chosen, due for the sake of keeping the time and keeping some time for a question, I've not addressed the question of membranes, because I'm not a specialist of this question. But if you extract organic matter from meteorites such as Murchison meteorite, some of the organic extract would spontaneously result in the formation of vesicles, as you can see on this picture. And this is a kind of spontaneous organization of organic compounds which are made of amplified chains. So one chain liking water, which is heading to the water, and the other face of the organic compounds, the tail, which is hiding from the water. So this is the reason why they tend to organize into vesicles. So it spontaneously occurs when you extract organic matter from some meteorite. You will be able to see the lecture given by Alessandra Rotundi on comets, but comets are also amazing objects that have been formed at the same time as the planets, and so far more than 20 small compounds have been detected from the Earth in the atmosphere of comets, and measurements that have been made in situ by the Giotto Vega spacecraft in 1986 during the exploration of the Halley comet. And after the sample returned by the Stardust mission has shown that the complexity of comets is similar to the one of the carbonaceous meteorite. And we are really living a exciting time, because in 2014, the European spacecraft, the ESA spacecraft Rosetta, will reach a comet and will actually land on the comet and make in situ analysis of the organic composition of the cometary grains. And we will learn a lot more on comets and the composition of comets in the next few years. So you may wonder, and you would be right to wonder, yes, but why? What is the origin of such a complex diversity in meteorites and comets? And then you can get back to the laboratory and build studies that, like Miller did in the 50s, like the one I just described on the hydrothermal vents. You can try to simulate the kind of organic processes occurring in the comets, in the ices, in the bulk ices that are the building block of the comet. And to do that, we simulate the cometary ice, which are linked somehow to interstellar ices. And we have experimental setups in the laboratory where we can cool at very low temperature, 10 kelvins, a sample holder, and on this sample holder you can condensate a gaseous mixture made of what you see are the main components of the comet. I mean, water, methane, ammonia, carbon monoxide. So we know those compounds are present in the comet. They are frozen on the comet at low temperature, and when they are in the interstellar cloud or in the outer regions of the solar system, those ices are submitted to energy deposition, and that kind of energy deposition can be UV from the neighboring stars. And so here you have the sample, you have the gas that are cooled at low temperature, and then you have a UV lamp that is simulating the energy deposition of the neighboring stars. If nothing happens, when you are done with your experiment, you turn off the cryocooler, the temperature is rising, and then water, methane, CO, ammonia would be pumped out from the sample holder. But it's a different story that is occurring. When you are back at room temperature on the substrate, something remains. And when you start to analyze that kind of material, you see that the compounds from the starting ice have been broken by the irradiation, and they have started to get interacting one with the neighboring one, and they have synthesized complex organic residue, which is still solid at room temperature. And when you get into the analysis of that kind of material, you are facing a molecular complexity which is dramatic, very hard. And here is a list of the kind. It's not a complete list, but the list of the kind of molecules which are synthesized during such simulation. So starting from an ice mixture, putting some energy in this ice mixture, you would result in the same kind of chemical factory as a primitive atmosphere which was made of reducing compounds. So you have any kind of organic compounds you would need, you would wish would be probably present in this list. And look here, in this list you have amino acids, but you have to pay attention to that kind of detection, because to get the detection of amino acids, you have to collect the residue and to put it in very harsh condition. You have to conduct an acid hydrolysis with concentrated HCl at high temperature, and it's only after this kind of process that you get the amino acid. So is that kind of processing acid hydrolysis relevant of what could have happened on the primitive Earth? Is some questions that have to be addressed, to be debated within the scientific community, but it's not there in the comet. Amino acids are, except maybe glycine, are not in the comet, but the kind of material, complex organic material present in the comet, once in liquid water, would result probably in the formation of amino acids. And again, as I showed you before, if you place this residue in water, you would observe spontaneous formation of vesicles that could be membrane or primitive membrane to the first living system on Earth. So if I want to summarize both endogenous sources and exogenous deliveries of prebiotic molecules, when it turns to the contribution of the atmosphere, it's a very interesting field, but it depends on the starting mixture, and I'm not sure we will be able at some point in the science history, in our knowledge of the Earth, to be sure 100% that the composition of the Earth atmosphere was this one or this other one. The contribution of hydrothermal vents can produce and destroy organics. We still lack a good set of experimental data to fully investigate this question. And exogenous delivery, which are still an ongoing process and which give results every year, I would say. And I'm not talking about those three items to try to have one winner. The question is not to say this is the atmosphere, these are the hydrothermal vents, this is exogenous delivery, which were at the origin of life. For a chemist, I don't care. Too many cooks can't spoil the prebiotic soup. We need that starting material, no matter it comes from the atmosphere or comets. But now, the key question for the prebiotic chemistry are not at this stage. The key questions are to understand where and how we can get beyond this stage of complexity. And this is what I'm going to address to conclude this lecture: how we understand, how we study this link from simple to complex compounds, but not the kind of complexity like proteins and DNA. I won't be able to say much about this. So I'm going to just give you ideas. I know some of you are not chemists, so you won't be eager to listen to hundreds and hundreds of mechanisms, but I want to give you some input, some ideas of what is well established and what is not that well established. And so for the synthesis of amino acids, this is something that is going back to 1850, the Strecker synthesis. So Strecker, which was a German chemist, was not studying the origin of life, but he was one of the first trying to synthesize amino acids in the lab. And he figured out that mixing an aldehyde, ammonia, and hydrogen cyanide would result in the formation of amino acid. You started mixing an aldehyde with the ammonia, and then you will produce what we call an imine. And then this imide would react with HCN. You have here the alpha amino nitrile, and if you have the alpha amino nitrile in liquid water, the nitrile function will be hydrolyzed and turned into acid carboxylic. And there you have an amino acid. Amino function, acid function, you get your amino acid. It's not a big deal to understand how you can get amino acid in the Miller experiment or on the primitive Earth. This synthesis, I really love it because it's a synthesis of adenine. Adenine is a nucleic base in the family of purine, and adenine is the letter A of the genetic alphabet. And it turns out that if you want to synthesize adenine in prebiotic conditions, so this is not how it happens in our body, because synthesis of adenine in prebiotic conditions starts from HCN, which is a very poisonous compound. But it's the polymerization of HCN, dimer, trimer, tetramer of HCN, photolysis, so some photons, which will reorganize the molecule, again, photolysis, and then a fifth HCN molecule, and then you get the adenine. So it's extremely, extremely simple to understand how adenine might have been synthesized on the primitive Earth, or why it's not stupid to imagine there is adenine in comets, or why we can detect adenine in meteorites. So those kind of nice syntheses have been studied after Miller-Urey experiment, because chemists have really started to try to understand how to get those bricks, what we call the bricks of life. We also need to account for the formation of sugars. Remember, to get a DNA or RNA molecule, you need sugar, and it's also something that is back in the 19th century, because a Russian chemist, Butlerov, figured out how to synthesize sugars from very simple compounds such as formaldehyde. And this synthesis starts from formaldehyde with a suitable catalyst such as calcium hydroxide, calcium carbonate, or clay minerals. And when you make this mixture, you will get polymerization of formaldehyde, and here you will get glycolaldehyde, another formaldehyde, glyceraldehyde, and if you mix glycolaldehyde and glyceraldehyde, you would get a pentose sugar, which is actually ribose. Desoxyribose are pentose sugars, and you can get sugars with 6 atoms. And so this is a very complex chemistry, and it's one of the problems of the origin of sugar, because when you do that kind of synthesis, you don't select pentose sugars, you don't select ribose, desoxyribose, you have a mixture of many, many sugars. And this is a chromatogram of what can be analyzed after the kind of mechanism I've just described. This mechanism results in that kind of diversity of compounds. Each peak is a different sugar. And you see, if you are looking for ribose, ribose is only one peak among this large variety of sugar. So why life did selected this sugar? Is it really that kind of synthesis that really led to the formation of sugar? And that is a critical question, and a lot of people have been wondering for years and years about the origin of sugar. And then when you get amino acids, nucleic bases, sugars, you need to link them together. And it's nothing but simple, because it can occur every time in any kind of living being. You have the ribosome, and ribosome is a complex structure to link amino acids one after the other according to the information transmitted by DNA and RNA. But if you want to do it on a natural matter, it's very hard, because if you want to link two amino acids, you have that kind of reaction. And this reaction releases water. The formation of a peptidic bond is the release of a water molecule. And if you are familiar with chemical equilibrium, if you have something in large amount on one side of the equilibrium, this will be pushing the equilibrium one way or the other. And if the condensation of amino acid is occurring in water, you see, you are in water, then the condensation will be very difficult, because you are producing water, you are in water. It's not a good way to proceed for this synthesis. So there are a lot of studies on catalytic synthesis on surface minerals to try to get rid of this problem of water excess. And if you want to synthesize polynucleotide, I mean, adding the nucleic base, adenine, for example, to ribose. So you have a good way to synthesize adenine, you have a good way to synthesize at least some ideas to synthesize ribose, and then you want to link one to the other. And it's not working. You need to be in the solid state, you need to be at high temperature, and the efficiency is only 3%, and it's not at the right place. It would be added somewhere on the sugar, but not on the specific place it's meant to be added. And so this is the production of what we call a nucleoside. And if you want to add a phosphate plus nucleoside, it's a nucleotide. Then again, you need heating, and the phosphate won't be added at its natural place. So there's been a lot of work, a lot of thinking, and only a few years ago, a breakthrough. I really think it's a breakthrough has been done on this question. It's been published in Nature in 2009, and it's a very elegant chemistry by the John Sutherland team in England. So this is the classical way. You want to synthesize the sugar from the glycolaldehyde and glyceraldehyde, and you have the sugar, and you want to make the nucleic base. Here is a cytosine from small constituent, and you want to add them together, and it's not working. And the new idea that has been published by this great team of prebiotic chemists, and I think they are really thinking out of the box, is to bypass this complex, this simple addition of one brick to the other. They bypass it because they are following the green path, and they are not forming the sugar, the nucleic base. They actually start from glycolaldehyde, glyceraldehyde. Here is cyanamide, urea, cyanoacetylene, cyanoacetaldehyde, and they mix them together. They use phosphate as a catalyst, and then they are following the green arrows. The chemist would be probably wanting to read the paper in more detail, but then following the green arrows, they synthesizing the nucleotide from scratch, from the most simple compound they get to the nucleotide. They don't add pieces to get together like prebiotic chemists have been trying to do for years and years. And so I think this is a real breakthrough, because it's not the solution for the origin of life, but the chemists have been starting to think about prebiotic chemistry on a different way, and this is very interesting, because for years we've been thinking on the origin of life as a linear process. As I showed in the beginning, you start from simple to more complex to more and more complex, and then to life. But maybe the solution is in some bypass, some complex material that could, in the end, result in some kind of simplification when it's added in liquid water. And that kind of complex mixture of carbon, hydrogen, nitrogen, oxygen is the kind of mixture you find in the solids, the organic A's on Titan, in what we call the yellow stuff. The yellow stuff is the organic residue when you make irradiation of ice, simulating the chemistry on interstellar ice and cometary ice. You may remember the brown matter coating the bulb of Stanley Miller. It's also made of this complex, of that kind. It's not the same, but that kind of complex and very hard to study organic material. if you let HCN polymerize, you can get some adenine, as I showed you, but you also get a brown and dark matter made of large chain of organic carbon, of organic compounds. And if you extract all the organic components of meteorite, most of it is insoluble in water and is made of long chains of carbon, oxygen, hydrogen, and nitrogen. So these are pictures of this kind of matter. This is Stanley Miller. This is a brown coating. This is Mario Greenberg, who was actually the one who invented the kind of experiment simulating ice chemistry. And this is Clifford Matthews, who has been studying for years the chemistry of HCN. And this is a mixture of HCN after 1 day, after 1 week, and you get spontaneously this brown and complex organic material. And so this is a classical way to get adenine, as I showed you previously. And this is another hypothetical way, but I think it's worth to dig into that kind of direction. This is a representation of HCN polymer. And within the HCN polymer, if you put this HCN polymer in water, you can get adenine. So you would get from simple to extremely complex, but not really relevant to life origin, to complex in sense that it's relevant for life origin and maybe not as a linear chain as it's been thought before. I'm almost in the end. Just another recent result I want to mention. I'm going back on Titan, and this is a kind of experiment conducted in the laboratory I'm working with François Rollin. This is a simulation of Titan atmosphere, a spark discharge in a mixture of nitrogen and methane. And this is the kind of solid residue you get after this spark discharge. And if you take those aerosols analogues made in the laboratory, if you put them in water, in liquid water, and sometimes mixture of liquid water and ammonia, you get from this mixture amino acids. But as I told you, if you have amino acids, you don't have life. You still have to figure out how to connect amino acids to get proteins. And this is for me only the beginning of the story. Amino acids are the beginning of the story. So it's time for some concluding remarks. And I'm going again to quote Alexander Oparin, because you may not know, but he wrote 2 versions of his book called The Origin of Life. In his first version in 1924, this is the conclusion. In 1924, he said, "We have every reason to believe that sooner or later we shall be able practically to demonstrate that life is nothing else but a special form of existence of matter." Some promotion of Soviet biology, then the successes scored recently by Soviet biology hold out the promise that the artificial creation of the simplest living being is not only possible, but that it will be achieved in the not too distant future. So in 1924, he was very optimistic. He thought very soon we would have artificial origin of life in the laboratory. But he wrote a second version of his book in 1938. It was also called The Origin of Life. It was a thicker book with more ideas. And this is the conclusion. The end of the book, he says, we are faced with the colossal problem of investigating each separate stage of the evolutionary process as it was sketched here. The road ahead of us is hard and long, but without doubt, it leads to the ultimate knowledge of the nature of life. The artificial building or synthesis of living things is a very remote, but not an unattainable goal along this road. So within 14 years, you realize that it was not that simple. We are still facing that kind of gigantic complexity. And exploring the solar system, connecting this exploration to work done in the laboratory by astrochemists and by actual chemists who are bringing up new ideas, like it's been done recently, are promising, but I can't promise you that within 10 or 20 years we will have the answer to the question of origin of life. But I'm certain that in interdisciplinary approach like the one conducted in astrobiology will be the good path to achieve this goal. So I thank you for your attention, and I'm ready to answer your question.
Wonderful. Thank you very much, Hervé. If I may start the question session myself, actually, I do have a couple of comments that perhaps you can address. My first is with regards to the Miller-Urey experiment. Perhaps you could comment on the quantities and the chirality of the amino acids that were produced at the time.
So the yield is a few percent of carbon that was in the methane, and a few percent is found in the final organic mixture. So that's quite efficient, and the chirality is yes, I've not talked about it, but it's a racemic mixture. So when you do the kind of synthesis either by the Miller-Urey experiment or by irradiating ice or simulating hydrothermal vents, each time you have a racemic mixture, which means you have the same amount of each configuration of the amino acid.
Okay, and as you know, there's also currently a Miller-Urey experiment being developed for research on the ISS. Perhaps you could make a comment on the cleanliness of the apparatus before the experiment starts. Presumably in 1952, it was pretty difficult to make sure that the equipment was amino acid free to start off with, and how that would possibly influence the outcome of the experiment.
Yes, the one critical issue when you try to detect amino acids in an experiment is that you don't want to detect amino acids which were there. Actually, we are full of amino acids. You touch something, you leave amino acids. So there are a few guidelines to try to figure out if you have contamination or actually amino acids that have been produced in your experiment. So one thing is racemic. If you have a contamination of amino acids for a long period of time, you will have an excess, a large excess, if not 100%, of the L configuration. So if you find in your experiment only L configuration and only amino acids that are part of the 20 members list of the living system, it means that it's definitely contamination. During the Miller-Urey experiment, he was not able to discern chirality, but he detected glycine, and he also detected other amino acids which are not in the what we call the proteogenic amino acids. So they were definitely not contamination. Nowadays, when we are facing that kind of questions, we start in the initial mixture, in the initial ice mixture, and in the initial atmospheric simulation. You use carbon which is marked with isotopes. So you're not using methane with C12. If you want to make sure that you synthesize amino acids which are not from contamination, you start with methane which is C13. And then if the amino acids you get in the end are C13, it means this is not contamination. So for the Miller-Urey experiment that is planned on the space station, so this experiment is try to study the influence of microgravity. They want to make a gaseous mixture interaction with the minerals. In microgravity, they plan now to start with each compound of the starting mixture will have a dedicated isotopic signature. So yes, cleanliness is a big deal in those experiments, and isotopic labels are a great help to figure out if it's a contamination or not.
Okay, wonderful. Thank you very much. I will pass the questions. Are there any questions from University of Napoli?
Ci sono domande, ragazzi? No questions from here.
Okay, wonderful. Thank you very much. Tino, do you have any questions from Dresden? Tino Schmiel, can you hear me? No, can you hear me?
I'm Tino Schmiel's Erasmus student, and thank you. I don't have questions.
Okay, thank you very much. Let's go to University of Szczecin. Do we have any questions there?
Yes, so we have a question which is now coming. So please. So this is a question from Adam Watzner. I'm giving
Thank you very much for a nice lecture, first of all. My question is about energy sources. Are taken about under account other energy sources than lightnings or thermal vents? For example, meteorites impacts or natural radioactivity.
So yes, I've talked about energy sources, and I've been talking about the most obvious, or at least the one that have been the most extensively studied. So the sun, that's a major energy source, and the lightning. Well, of course, when there is an impact of an asteroid on Earth, the shock wave can affect both the composition of the atmosphere where the shock is done. So that's something that has been studied, but I'm not sure I'm well informed on this topic. But there are chemistry. If you have shock impacts in a mixture of small compounds, you will have synthesis, and you are also able to change the nature of the compounds which are in the original body. There have been studies of people, for example, they placed a mixture of amino acids in a gun bullet. They shot with the bullet with the gun, and it reached a surface, and then they made analysis of what was still on the bullet, and they studied that most of the amino acids did survive, and some chemistry occurred. And for instance, the kind of linking between amino acids I said were very difficult in water. That kind of linking between amino acids can occur after a shock. But I'm not sure it's enough to account for the origin of life. But there are that kind of chemistry also occurring. So people are also studying on the kind of source in a primitive body of the solar system, primitive comets, primitive asteroids, and what kind of temperature is reached when there is decay of radionuclide in the body. So there is work on these topics too.
Okay. Any other questions from the University of Szczecin?
No, thank you.
Okay. I have a little message from Natalia Gontareva, who informs me that there were no questions for Hervé. And unfortunately, they have to close their session now because they're 3 hours ahead of us. So we will not have them present for Franco's lecture. However, I do encourage that the students participate or ask questions in the YouTube discussion, and then we can try and address some of the questions there if they're burning, or again, I repeat myself, but if we can also have some questions at the end of the session in February. University of Turku, do you have any questions for Hervé?
Can you hear us?
Yes, we can hear you.
Okay, we have a couple of questions. First of all, when you measure the chirality of proteins, or sorry, amino acids, how do you do this technically? Is it a difficult measurement? And how can you make claims of extremely small values that are non-racemic? So one chirality dominant, say by a factor of 10 to the -6, like has been done in Mars meteorite.
So to measure chirality, you need an instrument which is chiral. So most of the time it's done by chromatography. You inject, for example, in gaseous phase or in liquid phase, a mixture of amino acids. And the principle of chromatography is that the molecules are interacting with what we call the stationary phase of the chromatographic column. And each molecule interacts in a different way with the stationary phase. And then they spend different time reaching the end and the detector after the chromatographic column. If the stationary phase is chiral, then the interactions of the molecules with the stationary phase will depend on the chirality of the compound. And so, if you browse a catalog of chiral chromatographic column, you will see that some are specific to chiral compounds because the stationary phase is chiral, has a specific geometry, and the geometry of the compounds will affect their interaction with the stationary phase. So it means that if you have configuration L and configuration D, configuration L won't have the same kind of interaction than configuration D, and for instance, configuration L will get faster and reach faster the exit of the chromatographic column than the other one. So you will have two peaks, two chromatographic peaks, and doing calibrations, you will be able to see, okay, we have L here, D here, and you can do quantitative measurements, and you can figure out if you have the same amount in both peaks, or if you have one peak which is in excess to the other. Of course, it depends on the amount of material you have for the analysis and the uncertainty you measure. You have to be very cautious. And the first people who claimed that there is an enantiomeric excess in some carbonaceous meteorite were extremely careful on their discovery, and they ran a lot of tests, and they made sure that they were outside of the uncertainty for the measurements.
Any further questions from Turkey?
Yes, yes. I would like to ask you, first, thank you for a very interesting lecture and quite new idea that the first organic molecules would have been very large complexes that then somehow took form of active building blocks of life. So I would like to ask you on what you think that what would be the suitable conditions for producing this kind of very large and very non-specific molecular complexes. Apparently it would require high concentrations of ingredients and then non-aqueous conditions and a lot of energy. So where would you produce this kind of large precursors?
The common thing between efficient synthesis in the atmosphere, efficient synthesis in simulated hydrothermal vents or in interstellar ice is that you have to, the starting components, the starting molecules have to be quite reduced. So as I said, if you mixed CO2 and nitrogen, so neutral composition in the gas, and you make energy deposition, you have almost nothing. It's the same thing in the ice. If you mix CO2 and nitrogen as ice, and you have no source of hydrogen in terms of water, of methane, or ammonia, you won't get anything. So the common, and in the atmosphere of Titan, you have carbon in the form of methane, so surrounded by hydrogen. So to get complex mixture of organic, you need to start with a somehow reduced environment with at least a good source of hydrogen. So that's the basic requirement. Either in the atmosphere or ice, or it's always the case.
Okay, thank you very much. No further questions.
Okay.